Talk about amino acids. Amino acid structure and peptide bond formation. When we think about an amino acid, we have a really, really simple molecule. We start with a nitrogen. Covalently linked to that nitrogen are two hydrogens. Covalently linked to the nitrogen is a carbon. Covalently linked to this carbon is a carbon with a double bonded oxygen and an OH. You'll recognize the carbon double bonded oxygen OH as a carboxylic acid. This is the same structure we saw on the fatty acid. Gives the fatty acid its acid characteristic. This is a carboxylic acid. This NH2 is said to be an amine. Hence the name of these structures as amino acid. This carbon now is also linked to something known as an R group. The R group can be 20 different kinds of side groups. It could be as simple as a hydrogen and glycine. It could be two carbons as in glutamic acid. It could be a ring structure as in phenylalanine. It could be a ring structure with another carbon in between, such as tyrosine. We're not going to get into the details of the molecular or atomic structure of these R groups. But let me say, though, that R groups come in 20 varieties. And these 20 varieties give rise to the 20 species of amino acids. Life on the planet is made of protein, and the protein is derived from simple 20 amino acids. And the ways we make different proteins depend upon how we string these amino acids together. But before we get into the R groups and talk about what the R groups are doing, what I want to do is do the nuts and bolts, walk through the nuts and bolts of how we're going to link one amino acid to the next. Because after all, when we think about the amino acid structure and the peptide bond, where we're headed is proteins are polymers of amino acids. Where we're headed now is to try to think about how we can make these polymers. Because what we're going to do is we're going to link one amino acid to the next, one amino acid to its neighbor through something known as a peptide bond. So what we're going to do here is we're going to try to think about what we mean by a peptide bond. So in blue is going to be another amino acid. As we started out with this morning, we can now imagine, again, a kind of dehydration synthesis. We can take the hydrogen from the amine and the hydroxyl from the carboxylic acid and generate this HOH. So that's the dehydration step. We've taken water out. And in the process of doing that, we're now going to take this carbonyl carbon, the carbon of the carboxylic acid. The carbonyl carbon is now going to be linked directly by a covalent bond to the nitrogen of the adjacent amino acid. This carbonyl carbon nitrogen bond, of course, has a name. It's called a peptide bond. We'll see a little bit later that these peptide bonds are formed in biology by the enzymatic activity of ribosomes, but leave that in the background for the time being. What we can do here is we can now then take this first amino acid, link it to the second one, and do the same out here, where again we have a nitrogen with the two hydrogens. We have the carbon with its R group. And we again have the carboxylic acid. We can follow through the same pattern, where here we have the hydrogen. We have the hydrogen from the amine. We have the hydroxyl from the carboxylic acid to make water. And we can again form this carbonyl-carbon nitrogen bond, that peptide bond. We can do this over and over and over again. I'll show you one more here, to a green one. So again, we have the hydrogen, the OH, and the formation of the peptide bond. Depending upon the R group, we could have 4 different amino acids here, or we could have 4 of the same amino acids. It doesn't matter. That's determined by the R group. What I want to emphasize, though, is that although we have laid out here 4 amino acids, we could lay out 400 or 4,000 amino acids, all linked one to the next by these peptide bonds. But one of the ideas I want to show you here is that because of the nature of linking the carbonyl-carbon to the adjacent nitrogen, doing this over and over again, that we always have a kind of orientation to the molecule. We always have at this end the so-called amino, or N-terminus. And at the opposite end of the molecule out here, we have the C-terminal that I'll often refer to as the carboxy. Terminal. This becomes important to us because it gives us a nomenclature, gives us an endedness of the molecule, and we can talk about the N-terminal and the C-terminal. We can talk about the amino terminus and the carboxy terminus. We can talk about an orientation of the molecule such that we're moving toward the N-terminus or moving toward the C-terminus. So this is a kind of nomenclature that provides us a means of describing the orientation of proteins. The other major idea here that falls out from this is that the peptide bonds between adjacent amino acids determine the primary structure. Of the protein. This primary structure, when we talk about the primary structure, what we're really talking about is the amino acid sequence. Now, the amino acid sequence determines the amino acids in the protein, and that amino acid sequence will determine the kind of protein we have. Yes?
Is it the peptide bonds or the R groups that determine the amino acid sequence?
So, The question is, what determines the amino acid sequence? And what I'm trying to describe here is we link one amino acid to the next by the peptide bonds. So it is the peptide bond that determines the sequence of the amino acids as they're linked covalently. The type of amino acid we link one to the next will be determined by the R group. So this could, depending on the R group, this could be a lysine, this could be an arginine depending on its R group. So the amino acid sequence is determined by linking them together by peptide bonds, but the amino acids in the sequence are determined by the R groups on those amino acids, which are linked by the peptide bonds. Does that help? Okay. Anything else? So now what we've done is we've strung out these amino acids linked one to another by the peptide bonds. And theoretically, we could choose each of these amino acids at random from among the 20 different amino acids and link them by the peptide bonds.